All right, moving along, we have DNA replication. And the idea is, DNA is a molecule of heredity. We need to be able to make copies to be able to pass down from generation to generation to generation. Our current model that we have is called the semi-conservative mode of replication. We have a parent molecule, you break it apart, and then you kind of fill out each side with like new pieces of DNA. And that makes two new strands, but half is old and half is new. So it's called the semi-conservative model. There was some experiments, though, done by Messelson and Stahl that actually looked at three competing models of DNA replication. One's called conservative model, the other one's called semi-conservative, and then dispersive. So we just went over the semi-conservative, but in the conservative model, you start out with the kind of original strands of DNA, then they kind of open up, but the original strand sticks together, and then another strand is like wholly new, and then that original strand sticks together throughout, and then you have other new strands that are made, like that are just wholly new, completely new. All right, that's the conservative model. We went over the semi-conservative model, but then you got this other one called the dispersive model. So you start out with your parent strand, and then instead of keeping one strand that's wholly new and another one that's like really like totally old, it's like each strand has like pieces of old and new within it, to make up each kind of successive DNA replication. So if you look at the dispersive versus semi-conservative here, you can see this is this darkly colored one. That represents the old piece of DNA that's still part of the new one. Whereas in the dispersive model, it's like we have old, new, old, new, old, new. So how do you study that, right? Well, they used heavy nitrogen. So nitrogen normally has an atomic mass of 14. This one has an atomic mass of 15. That's why it's considered heavy nitrogen. So what they did is they took some bacteria, grew it in heavy nitrogen, and that means that the DNA is going to be heavier. All right, and then so that DNA that's in this one, like the original DNA, is going to be heavier than whatever else comes along. All right, so grow the DNA in the nitrogen 15. All right, then you let it grow another generation in just regular nitrogen. Okay, you take samples at 20 and like 40 minutes. The idea is that bacteria under good conditions, they can have a generation every 20 minutes. If in the lab conditions, it's going to be at the right temperature. The bacteria is going to have the right nutrients, so that's not too out of the question. Oops. So what they did at the at the first 20 minutes, so it says like they basically took a sample, they centrifuged it, so that gets all the cells kind of in a pellet, and then they extract the DNA and get it can kind of hang in the column, as you can see here. So like the first generation was here, and then the second generation we kind of had these two bands of DNA in there. So if we look at kind of our conclusions, our predictions, and then kind of how it fits with the data. So if it was a conservative model of replication, you would expect like a really, really like heavy strand at the bottom after that first replication, and a light strand. Remember, so we're keeping all the DNA. All the old DNA. We're not keeping all the old DNA. All right. So right off the bat, we can get rid of the conservative model of replication. All right, semi-conservative. So we're not having super light. We don't expect to get a super light band. We don't expect to get a super heavy band. We expect to get one in the middle. Okay, because it's got light and heavy strands combined, or light and heavy pieces combined. But whoops, hey, get back here. Interesting enough, the dispersive model is the same way. All right, each one you expect light, heavy. So, so by virtue of the fact that that first replication in the nitrogen 14, it only gave us one band. That allows us to get rid of the conservative model, but it doesn't allow us to get rid of anything else. So after that second replication with the semi-conservative, you expect another medium band, but then a light band. In the dispersive model, you expect like a light medium band, but basically you only expect one band versus two. And what they ended up getting was two bands of DNA, a really light one and a medium one, versus one like I call it light medium or whatever. Okay, so this allowed them to conclude that, hey, our model of DNA replication is semi-conservative. All right, so let's go into like how this actually takes place, because it's pretty. I think it's kind of amazing. So we really need to focus on the actual enzymes that are doing this. And here they are. Memorize this now. Next. No. So helicase is an important one. It breaks apart the DNA helix. Single-stranded binding protein is not an enzyme, but what it does is it binds to a single strand of DNA and keeps it from kind of coming back together. Topoisomerase, I don't really go over it, but basically what that does is it relieves tension on the DNA and keeps it from like breaking. Keeps it from breaking apart by breaking it. Primase makes something called a primer, and the primer is like a small piece of RNA, which kind of gets this stuff jump-started. DNA polymerase 3 makes polymers of DNA. I'll explain like how that one is different than DNA polymerase 1. And then we have DNA ligase, which kind of joins together the sugar phosphate backbone in this process. So let's go get into this. So all of this starts, it doesn't matter if you're eukaryotic or prokaryotic, at a site of origin. So DNA helicase will come along, it'll find a site of origin, and it will start this process. And it actually goes in two directions. So if this is a site of origin, if this is site of origin, we're going to go to the left and we're going to go to the right. The same in the bacteria. If this is our site of, this is our site of origin, we're going to go to the left and we're going to go to the right. And like for a circular one, they just kind of go until they meet in the middle. You can see electron micrograph of that right here, and then here you can see multiple sites of origin on the DNA in a eukaryotic cell. So it would have been in here somewhere, because here's a fork, and here's a fork, there's a fork, there's a fork, fork. Whoa, there it is, up close and personal, I think you get the idea, okay? So helicase comes along, it breaks apart the DNA helix. Single-stranded binding proteins actually bind to a single strand of DNA. So here is helicase, it's breaking apart. I'm not focusing on topoisomerase, but here you can see it made a little nick in the sugar phosphate backbone to, like I said, to relieve the tension. But once the DNA helicase opens the stuff up, single-stranded binding proteins land on here to keep this strand and this strand from coming back together. And then primase comes along here, and it is making a primer. And that primer is going from 5 to 3. It's in the 5 to 3 direction. Okay, so let's take a time out here. It doesn't matter if it's DNA or RNA, all the nucleic acids are synthesized in the 5 to 3 direction. Whoops, take it back here. So that you'll notice like the template strand here is going 5 to 3, and then the DNA is or the primer is going the opposite way, because remember, DNA is anti-parallel, so it's going this way. Now, replication on the bottom strand here would actually go 5 to 3, so it would actually go the opposite direction. Okay, and that's the start of how we copy, or how we make copies of DNA. So just I'm gonna refresh your memory, remind you, whatever our primer is made out of RNA, and it's short sequence, handful of RNA nucleotides. Since this is not going to be in our actual final copy, we don't want a lot of RNA, but we need enough to kind of get everything started. Yeah, sorry, 5 to 10 nucleotides. So, DNA polymerase. This is going to come nestle up next to our primase, or I'm sorry, our primer, and that's going to get this whole thing kicked off. And it's interesting. Rate of elongation is 500 nucleotides per second for a bacteria, 50 in people. That's still a lot. Every second that's ticking off, it's like 50, 100, 150, 200, and so on. You know, you can get quite a few done in, you know, a minute, 2 minutes, so on. So I'm gonna get away from this and get right here. Get back to kind of our excuse me, our story. So DNA polymerase 3 comes along here and starts adding bases to our DNA strand. So if there's like on the top strand, if there's an adenine, DNA polymerase 3 is going to fit a thymine to it. If there's a cytosine on the top, DNA polymerase 3 is going to add a guanine to it. So it's just adding the complementary base pairs to or complementary base to whatever it's encountering. So DNA polymerase 3 just happily goes along and adds complementary bases. You can see here, you know, it would have started here with the primer, and then on the top strand, it's actually going into the fork, and it's just going to go until it runs into like another like double strand. Okay, it's going to follow on the top strand. It's going to follow DNA helicase. And in that case, we call that continuous replication. And the top strand here, we call this our it's our leading strand. But there's a lagging strand. So since there is a requirement, like we have to go from 5 to 3, on the bottom strand, 5 is on the left and 3 is on the right. So the helicase kind of has to open up some stuff, and then so it opens up some stuff, and then we replicate out. Opens up some more stuff, we replicate out. Open up more, and we replicate out. What the cell doesn't do is it doesn't like open up like the DNA and go all the way then, and then we start the replication. No, what it does is we lay down a primer and we go. Lay down another primer and go. Lay down another primer and go, and so on. One of the people that kind of was helping determine this and kind of figured it out was a man by the name of Okazaki. So here we go. So you know, we notice, okay, we got laid down a primer on our lagging strand, that bottom strand, and then DNA polymerase 3 comes along, and we make like a copy. Well, Okazaki figured out this, and these little fragmented pieces of DNA are called Okazaki fragments. All right, so to the left of what we have over here, we're going to lay down another primer, and then polymerase 3 is going to come along and do the same thing like we got here. So here's our primer, DNA polymerase 3. It's going to go until it hits this primer, and it's going to fall off. Although you don't see it falling off. But here's the problem. A couple problems. A, we still have we have like this RNA kind of in this mix here, and we don't want that. We don't want that at all. So DNA polymerase 1 actually has to come along and goes up here, and it's like, hey, there's RNA. Let's edit it out. Chop, 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 and then adds in the right DNA. All right, and then, that's great. We got rid of the DNA, but you can kind of see right here, there's a nick in our sugar phosphate backbone. We don't want that. We want to get rid of it. So ligase comes along and seals this up. And so if you think about like the term ligase, probably doesn't really mean a lot. ACE is going to tell us it's an enzyme. But if you think like ligament, ligature, these are all things that kind of tie things together. And that's what DNA ligase does. It ties our sugar phosphate backbone together here. Excellent. And then so it'll seal it up, and then we're good to go. Some interesting. Whoa, get back here. Some interesting kind of ideas on how DNA polymerase 1 and 3 edits. So here's DNA polymerase 3. As it's going along, say it adds a thymine here, it goes forward. It actually looks back to see if it put the right base in. And if it didn't, it'll edit it out, chop, and put the right one in. And it goes here, adds another DNA base, looks backwards. Hey, is that right? Yep. Okay, move along, move along. But if it does make a mistake, it has the ability to fix it. Same with DNA polymerase 1 down here. It starts to, but it actually can edit forward. So it's like, hey, there's RNA, let's get rid of it, chop, put in the DNA, and then it'll move, forward, and it looks backwards. Hey, I put the right one in, or oh, I didn't. We got to fix it. So polymerase 1 can edit forwards and backwards. Polymerase 3 only edits what it laid down. Woo! So here it is in all of its glory. You can see the leading strand on top and how we have like continuous replication. We have the lagging strand on the bottom where it's not continuous replication. And you can see we've got our different like enzymes, whether it's primase, DNA polymerase 3, DNA polymerase 1, or ligase. All right, and that's our semi-conservative model of replication. Yeah, this is just kind of, if you're interested, this has got, it's showing us how, some of this stuff is edited. And I will say, like, if it is kind of put in wrong, the DNA will kind of bow out, like you can see up here, because the hydrogen bonds aren't being made quite right. So, we do have that ability to kind of say, hey, this isn't fitting right. We got a square peg and a round hole. Let's chop it out.